have used a previous demonstration to show you a simple lecture. Now let's look at a concept that is much more complex. This example comes from my lectures on biotechnology. The concept I will present is called PCR. PCR stands for polymerase chain reaction and is used to amplify selected DNA sequences, much like a Xerox machine makes many paper copies from one sheet of paper. These DNA copies can be used for sequencing, diagnostic identification, and cloning genes from one organism to another. So let's watch this short movie on how the polymerase chain reaction works. I have a few comments about the process once the movie is completed.
The polymerase chain reaction, or PCR, is an alternative to cloning for isolating a DNA sequence. PCR can selectively amplify a single copy of a desired sequence that is present in a complex mixture of other DNA molecules. In this example, a mixture of genomic DNA molecules, shown as blue ribbons, contains the sequence we wish to amplify, called the target sequence, shown in green. With PCR, we can make billions of copies of the target sequence without having to purify it beforehand. In the first cycle of PCR, the DNA molecules are melted by raising the temperature to 95 degrees. After the strands have separated, the temperature is lowered to 60 degrees so that a pair of short oligonucleotide primers, specific for the 3' ends of the target sequence, can bind, one primer on each strand. The primers then direct a DNA polymerase, called Taq, to synthesize the complementary DNA strands from free nucleotides. In the entire mixture of genomic DNA molecules, only DNA containing the target sequence is copied because the Taq polymerase can only copy molecules that have a primer attached. At the end of cycle 1, both strands of the genomic DNA molecule have been copied to form two partially double-stranded DNA molecules. In the next cycle, the same steps are repeated. DNA duplexes are again melted by heating to 95 degrees. Since Taq polymerase has the special property of being thermostable, it is not inactivated at this high temperature. The primers anneal to the target when the temperature is lowered, and the target strands are extended by the Taq polymerase. The temperature can be raised to 72 degrees to speed this reaction. At the end of cycle 2, 4 partially double-stranded molecules are produced. All contain the target sequence plus some adjacent DNA. During the third cycle, the DNA strands are again melted. The primers hybridize to the target sequence, and the Taq polymerase synthesizes complementary DNA strands. Cycle 3 produces 2 doubly stranded molecules that contain only the target sequence, as well as 6 longer molecules that contain the target and flanking sequence. In subsequent cycles, the number of target sequence molecules will increase exponentially. At cycle 4, the DNA strands are melted, the primers anneal, and the DNA strands are copied as in the previous cycles. Eight copies of the target molecule and 8 longer molecules are produced. In cycle 5, the heating annealing and synthesis steps are repeated. By the end of cycle 5, there are 22 target molecules and 10 longer ones. The number of target sequences continues to grow exponentially at each cycle. By the end of cycle 30, the target sequence has grown to more than 1 billion double-stranded target molecules, whereas there are only 60 copies of the longer DNA molecules. This means that the final product is virtually pure target sequence. Thus, like cloning, PCR selectively generates a large number of copies of a desired DNA sequence from the original mixture of DNA sequences.
First, let me state that the gene sequence that is copied is dependent upon which primers you put in the mix. If you want to copy one gene, you put in one set of primers. If you want to copy another gene, you put in a different set of primers. All the DNA between those two sets of primers will be copied and will be the target gene. Therefore, you can copy almost any gene from any organism. Where do you get the information to design the primers for your specific gene of interest? That information can be found in a place called the Gene Bank at the National Cancer Institute. There are genome data from thousands of organisms that have been sequenced, and that information is deposited in the Gene Bank and can be used for designing primers. So what can you do with the copies that you made from the PCR reaction? Well, let's take a look. Well, the first thing you can do is you can sequence your PCR product to see if those genes are identical with those of other organisms. Sequencing determines the order of bases in a molecule of DNA. Instruments usually perform this task. For example, this ABI sequencer here, and the results you get, for example, are a sequence like AA, GT, CG, CTT, and this will go on for sometimes hundreds of thousands or millions of bases. Every organism's genes are different, and therefore you can use them to identify them. You can also use this information to test for mutations, like, for example, changes that make a bacteria resistant to drug therapy. This is most important when you're treating infections or human diseases. You can even use the sequences to determine how the bacteria became drug resistant in the first place. You can also use them to determine, for example, what mutations occur that turn normal cells into tumor cells. And you can use the sequences in a technique called DNA fingerprinting. DNA fingerprinting is a molecular technique that can be used to determine if two samples of DNA came from the same source. This is a technique they use to prove people innocent of crimes or possibly guilty. For any particular gene sequence, only about 1 in 10 million people have the same identical sequence. There will be minor variations which DNA fingerprinting can detect. You can also take this PCR gene product, which is pure, and use it in genetic engineering or cloning. You can take the gene copy, insert it into a plasmid, which is a very small circular piece of DNA, and insert that plasmid into a bacteria like E. coli. You can grow this new clone, and it will express the protein of the gene you have put in the plasmid. So, for example, we can clone things like plasminogen activator, insulin, erythropoietic protein, interferon, epidermal growth factor, human growth hormone, and even vaccine components like those used in the hepatitis B vaccine. Cloning is an extremely valuable tool in biotechnology. A similar technique to PCR is called RT-PCR, which stands for reverse transcriptase PCR. It's used to study gene expression. An enzyme, reverse transcriptase, is used to make a DNA copy from messenger RNA. Messenger RNA is present only if a gene is turned on. If the gene is not turned on, there will be no messenger RNA. The DNA copy created from the messenger RNA can then be amplified using traditional PCR to see which genes are turned on and which are turned off under various circumstances. This technique is used to study diseases and how we can fight them. A perfect example is cancer. In a cancer cell, which genes are turned on that are not turned on in a normal cell? How does this difference make the cancer cell grow rapidly and uncontrolled? These can be answered through RT-PCR. Real-time PCR is another technique which is based upon the PCR technology. It is similar to PCR, but in addition to the two specific primers, there is a specific probe that contains fluorescent molecules attached to it. As the DNA is copied and amplified, this probe is chewed up and destroyed and releases the fluorescent tag. The signal increases over time and can be monitored in what we call real time. The more template you start with, the sooner the signal becomes strong enough to break a threshold. It can detect as little as 10 copies of DNA, and you can multiplex this. You can use different colors of dyes in one reaction. At one time, you could only use 4 different colors, but today you can use up to 6. So what advantage does this multiplexing give us? Well, let's look at a real-world example. This technique was developed in our laboratory at CDC. STDs usually come in 2 forms. There's one group that are called urethritic diseases, and they include gonorrhea, chlamydia, mycoplasma, and ureaplasma. And the second group are genital ulcer diseases such as syphilis, chancroid, herpes 1, and herpes 2. So we can take a specimen from a patient, extract the DNA from that sample, and run a multiplex real-time PCR and determine which of the 4 diseases in each category they have. In some patients, they may have more than one infection, which we call a co-infection. In GUDs, they could have syphilis, chancroid, herpes 1, or herpes 2. With different multiplex real-time PCRs, we can determine which of the 4 they are infected with. This process condenses days and weeks of lab work into 2 hours. And we don't have to grow the organisms, which sometimes they fail to grow. But remember, we can detect as few as 10 copies of the DNA. So even with weak or mild asymptomatic cases, we can sometimes detect the presence of these bacterial pathogens because of this molecular technique. The end. I hope you've enjoyed this demonstration of how I integrate movies, pictures, information, and real-life applications to help students understand very important concepts. Thank you.